Hello everyone, welcome back to another video on the Education Always channel. Today we will be looking at stem cells. In this video we will cover stem cells in humans and plants, cell differentiation, cloning in plants, using human stem cells and therapeutic cloning. So, stem cells are cells that have not undergone differentiation. The cell which has not yet become specialised is called undifferentiated. An embryo develops from a fertilized egg. Cells at the early stages in the development of the embryo are stem cells. If cells are removed from the embryo, called embryonic stem cells, they will differentiate into any cell type. Some stem cells remain in the bodies of adults. These are adult stem cells. Adult stem cells are found in limited numbers at certain locations in the body. Adult stem cells can be found in several regions of the body, including the brain, eyes, blood, heart, liver, bone marrow, skin and muscle. Adult stem cells can differentiate into related cell types only. For example, bone marrow cells can differentiate into blood cells. Cell division in plants occurs in regions called meristems. Cells of the meristem can differentiate to produce all types of plant cells at any time during the life of the plant. The main meristem are close to the tip of the shoot and the tip of the root. In a growing shoot, new cells are being produced continuously near the tip. As the cells become older, further away from the tip, they become differentiated. They enlarge and develop vacuoles. Animals and plants produced by sexual reproduction begin life as a single cell, a fertilized egg. This cell must divide to produce a multicellular organism. It must also differentiate so that its cells develop features that enable them to fulfill specific roles. On the screen is a table of all the specialized cells and their job. This is the list for humans, and next up will be the list for plants. And as promised, this is the list for plants. Cells from meristems can be cloned. The cells are removed from a plant and grown in tissue culture. These cells are grown in a culture medium that contain agar to provide support and water for the grown cells, along with nutrients and plant hormones to promote cell division. Clones will be genetically identical to the original plant providing the meristem cells. This is useful to provide crop plants for large-scale cultivation that have desirable characteristics such as disease resistance. Producing new plants by cloning is quicker than allowing plants to reproduce and collecting and sowing seeds. It's therefore an effective way of producing new individuals from rare and endangered species, helping to preserve the plant. Stem cells can divide to produce new cells which can then divide into different cell types. They therefore have the potential to be transplanted into patients to treat medical conditions and disease. They could be used to replace cells that have been damaged or destroyed. For example, in type 1 diabetes, in cases of multiple sclerosis which can lead to paralysis, or in cases of spinal cord or brain injury. Embryonic stem cells can differentiate into a wider range of cell types, but they are difficult to obtain and their use raises ethical challenges. The best source is the 5-day-old embryo. Adult stem cells will differentiate into a narrower range of cell types. Therapeutic cloning could produce stem cells with the same genetic makeup as the patient. The technique involves the transfer of the nucleus from a cell of the patient to an egg cell whose nucleus has been removed. Regardless of their potential, there are multiple social and ethical and clinical issues with the use of stem cells that you should do your own research on. On the screen is the process of therapeutic cloning. And that's it for today. Thank you everybody so much for watching. Please don't forget to like and subscribe for more, and any questions, pop them down below in the comments. Thank you, bye!